on page B11, so we've been talking about how sugar is broken apart to make ATP. And we wrote on B11 the catabolism of fats and proteins for energy. Remember, the word catabolism means to break apart. Like in a catastrophe, everything breaks apart. Or cats rip up everything. All right? So we want to now focus on fats and proteins and how they're used for energy. We wrote that in order to make ATP, your cells, your body breaks down mostly carbs, mostly sugars. That's the favorite food that cells use to make ATP. Some fats and small amounts of protein. So what we're going to be learning is how fats and proteins are broken apart and used also as a source of energy. This is actually shown on the next page, B12. So here's another second scary drawing. In this second scary drawing, which is very similar to the first actually, what this is showing is how proteins can be used for energy, how carbs can be used for energy, and how lipids or fats can be used for energy. What we're going to be learning is actually the way fats and proteins are used for energy is they're going to be turned into sugars. So you can see there's arrows here, and it actually points here, and arrows here, and this points here. So the way we actually use fats and proteins for energy is they're turned into sugars. But just before we take a break, let's just review this part on what happens with sugars, because this is what we've already been learning. And then after the break, we'll look at fats and proteins. So first, glucose. All right, now sugar. Sugar's in our bloodstream. We have learned that sugars, glucose, can be stored as glycogen, right? And that process of storing glucose as this polysaccharide called glycogen is called glycogenesis. What's the hormone that promotes or causes that to happen? Insulin. Insulin is what makes it be stored as glycogen. And when we need to increase the amount of sugar in our blood, because it's too low, then we can break down some of those glycogen stores back into glucose to raise the blood sugar level back up. That's called glycogenolysis, the breaking apart of glycogen into glucose, and the hormone that promotes that is glucagon. Now, the glucose is in our bloodstream. It's going to be used by the cells of our body, and we know that glucose is first going to be split in half into 2 pyruvic acids, which are also known as pyruvate sugars. The process for splitting a glucose in half into 2 pyruvate sugars or pyruvic acid is called glycolysis. Don't confuse the word glycolysis with glycogenolysis, because that's why I'm you're bringing your attention to it so you don't make that mistake. You shouldn't, because the words are pretty clear. In glycogenolysis, it has the word glycogen in it, and lysis, to break apart glycogen. Glycolysis doesn't have the word glycogen, it says glyco, which means glucose. So splitting glucose apart is glycolysis. We have learned that once glucose is split apart into 2 pyruvate sugars or pyruvic acids, one of 2 things happens. If there is a lack of oxygen, the pyruvate sugar is reversibly turned into lactic acid. This is called fermentation. When oxygen becomes available, it'll be turned back. Assuming oxygen is available, it goes through, and here's that dashed line again. Here's the dashed line. It will go into the mitochondria, and there it will first undergo the transition reaction, and then the Krebs cycle, and then the electron transport system as the sugars are broken entirely apart into carbon dioxide and water. So in the transition reaction, if oxygen's available, the pyruvate sugar is turned into acetyl sugars. In the Krebs cycle, the acetyl sugars are totally broken apart into carbon dioxides. The hydrogens, of course, are largely picked up. They are either picked up by the coenzyme FAD, or the hydrogens are picked up by the coenzyme NAD. NAD is niacin, FAD is riboflavin. And these hot potato hydrogens and electrons are then transferred from one coenzyme to the next. That's that row of people, or coenzymes. Notice the very first coenzyme I mentioned parenthetically is coenzyme Q or Q10. Right? Take a look. Go to the drugstore and you'll see, or the market and look for it. You'll see. Anyhow, and we said that as these hot potato hydrogens are transferred from one coenzyme to the next, energy is being released to phosphorylate an ADP into ATP, to phosphorylate an ADP into ATP, to phosphorylate an ADP into ATP. So there's a lot of ATPs being generated here. And finally, the cooled down hydrogens and electrons, hydrogens are attached to oxygen to form water here at the end. What do we call this last process? Oxidative phosphorylation. We're phosphorylating, making ATP, and in the process, getting rid of hydrogens, and the oxygen acts as a hydrogen acceptor. What we want to now look at is how the cells of our body, how our body as a whole uses fats and proteins as a source of energy. And all this becomes really important in terms of what is collectively known as your metabolism, the biochemical reactions in our body. Let's first start by seeing how your body uses fats or lipids. So here on page B12, on the right-hand side, it shows fatty acids, and it shows there's an arrow, and it's pointing in both directions, so this is reversible, that fatty acids can be converted into triglycerides, or triglycerides can be broken back apart into fatty acids. Have we covered that before? Yeah. And so we know that, in fact, the fatty acids are stored in our fat cells by snapping 3 fatty acids onto glycerol to form a triglyceride. So let's say, how does our body use fats for energy? The triglycerides in our fat cells can be broken apart into fatty acids. I want to remind you that a fatty acid is just a long chain of carbon atoms with a bunch of hydrogens attached. Remember that? Remember this? That's basically a saturated fat, which is what we have in our fat cells, is saturated fatty acids attached to glycerol to form triglycerides. Now, what happens to these saturated fatty acids is they are split into pairs of carbons. They are split into pairs of carbons, 2 carbons each. It's interesting that fatty acids are always, they're chains of carbon atoms, and they are always a multiple of 2. So they can be 20 carbon atoms long, or 18 carbon atoms long, or 24, but they're always a multiple of 2. You will never see a fatty acid that's 21 carbons. It's always an even number, all right? So you split these apart, and what's a 2-carbon molecule? That's acetyl. Acetyl is 2 carbons. Did we cover that previously? Yes, we did. How did we represent an acetyl on this diagram? 2 carbons. So literally, by breaking apart a saturated fatty acid, it basically becomes a 2-carbon acetyl sugar. So once it's an acetyl sugar, it enters the Krebs cycle, and it's broken apart into carbon dioxide and hydrogen atoms that are picked up by NADs and FADs that are run through the electron transport system. So in other words, this fat, once it's turned into these sugars, is broken apart as if it had started out as sugar, right? It was just turned into it. This is incidentally a reversible reaction, and this actually explains how when you eat sugars, the sugars can be turned into acetyl sugars, and then they're snapped together into a fatty acid, and then stored as triglycerides. So we've certainly pointed out that if you eat a lot of sugar, you'll become fat. You form a lot of fats, and they're stored in our fat cells as triglycerides. Now, since a triglyceride is made up of 3 fatty acids, and each fatty acid could be 18, 20, 22 carbon atoms long, if it's split apart, it forms a massive amount of acetyl sugars very quickly. And whenever we're breaking apart fats very quickly, it forms so many acetyl sugars all at once that it kind of floods the system. And these acetyl sugars are then turned into something called keto acids. So in other words, if acetyl sugars, that's what these are formed faster than they can be funneled through the Krebs cycle in that cell, some of these acetyl sugars are turned into something called keto acids or ketone bodies. We're going to have more to say about that, but here's the bottom line for the moment. Whenever we're breaking down fats at a fast rate, we will form what are called keto acids in the process. Let's summarize what I've just told you by looking back on B11. So back on B11, here's what we wrote. Fatty acids can be reversibly converted into acetyl sugars and then broken down to release energy that's used to make ATP. We'll remember, and those of you who've had a nutrition class learned, that each gram of fat contains twice as many calories of energy as either a gram of carbohydrate or protein. Here we wrote that whenever there is increased catabolism or breakdown of fats, you form keto acids or ketone bodies. And there are a number of keto acids, and one of them is acetone. And we'll have more to say about acetone. Acetone has a kind of fruity smell. They used to use, the prime thing they used to use for nail polish remover was acetone. Now they've now got some new formulations that are not acetone, but if you remember, classic nail polish remover had a slightly sweet or fruity smell. They also, if you took a chemistry class, usually they use acetone in the chemistry lab, and it kind of has a sweet or fruity smell. Why that's going to be relevant is we've just told you whenever you're breaking down fats at a faster rate, you're going to form these keto acids, including acetone, and you can actually smell it on the person's breath. And we'll have more to say about that. Now that we've told you how fats are converted into sugars and then used to make ATP, let's look at how proteins are converted into sugars and used to make ATP. So on page B12, all right, now proteins. So proteins, if they're going to be turned into sugars and then broken apart to make ATP, first they're split apart into amino acids. And then something called deamination has to occur. You'd say, what's that? That's the removal of the amino group, of the amino acid. Now to help you understand what that is and why it has to happen, let's look on the bottom of page B10. So on the bottom of page B10, all right, let's remember what an amino acid looks like. We had learned last week that an amino acid has this, all amino acid, and there are 20 different types, have this basic structure. It begins with a carbon atom. On one side is an amino group, right? Nitrogen and 2 hydrogens. On the other side is the acid group, thus the name amino acid. Here in this position is always a hydrogen. And how the 20 different amino acids differ from one another is what's attached up here in the R position for the rest of it. Now, sugars like glucose, right? Glucose was C6H12O6. So sugars contain carbons and hydrogens and oxygen. Is everybody okay with that? Fats are mostly carbon and hydrogen. They have some oxygen, mostly carbon and hydrogen. But amino acids are carbons and hydrogens and oxygens and, nitrogen. If we're going to turn an amino acid into a sugar, we've got to remove the nitrogen from it. By removing the nitrogen, it will be chemically like a sugar. Sugars are carbon, hydrogen, and oxygen. This is carbon, hydrogen, oxygen, and also nitrogen. So the nitrogen has to be removed. So this amino group is removed. That's called deamination. De means to take away. Notice that when you remove the amino group, we'll come back to what happens to it in a moment. It's still, you still have the acid group, don't you? But it's no longer going to be called an amino acid if it doesn't have the amino group. It's called a keto acid. So a keto acid or ketone body is formed when you remove the amino group from an amino acid. Now there are many keto acids. And that's because there are 20 different types of amino acids, and when the amino group is removed, it forms different types of keto acids. The keto acids include acetone, acetoacetic acid, and beta-hydroxybutyric acid. All of these are mentioned in your book, but I specifically, especially want you to know acetone. It's easy to remember, and it becomes very important clinically, as we'll see. Before we see what happens to the keto acid, basically the keto acid is going to be turned into sugar. But before we see that, let's see what happens to this amino group. The amino group is turned into ammonia. This is occurring in your liver. Now, you'll notice, so NH2 becomes NH3. You know, and an obvious question is, well, like, where did it get its extra hydrogen? It's actually provided by water. Water provides a source of hydrogens. We're not going to worry about it, but don't worry about where it comes from. We can show you a big, complicated biochemical diagram showing the details. And then this ammonia that's formed in the liver is then converted in your liver into something called urea. Now, what's urea? Urea is really just two of these amino groups, or ammonias, that are kind of joined to a carbon and oxygen. That's called urea. Now, I don't care, again, so much that you memorize that structure. Not that it's that difficult. But I want you to know what urea is. So, urea is formed in our liver, and then it's released into the bloodstream. It's released into the bloodstream. It's a waste product. And we can measure this urea level in our blood. When they measure the amount of urea in your blood, which is really a waste product formed from deaminating amino acids, they call that the blood urea nitrogen level, or BUN level. Has anyone ever heard that term, BUN? You've seen that in the hospital. It just stands for blood urea nitrogen. Obviously, they call it blood because it's in the bloodstream. They call it urea because it's urea, and it's got nitrogen in it. That's why we removed it off the amino acid, so it's called blood urea nitrogen. It is the major organic waste product in our blood, and then our kidneys filter it out of the bloodstream. Isn't that the role of our kidneys, to filter blood? And it's excreted in our urine. And it is the major organic waste product in our urine. So this major organic waste product in our urine is formed whenever we're breaking apart proteins. Now, we're always breaking down small amounts of protein, but sometimes we start to break down higher amounts than usual, increasing the amount of urea in the blood. Now, if you've never heard of urea, or BUN, you'd say, I never heard of it, I don't even know what you're talking about. So let's help you understand it. We're going to look at the yellow cover, and we're going to look at the inside front cover of the Physiology lecture outline. And what I've done here is I've just reproduced some lab forms from local hospitals. So the top one is a blood test form used at Brotman Medical Center right on Venice Boulevard in Culver City. And here they can measure the sodium levels in the blood, the potassium levels. We're going to be learning how to read all this as we go on. I want to draw your attention right to here where it says urea nitrogen. So this is indicating the normal amount of urea in our blood, right? 5 to 25 milligrams per deciliter. So they'll call it urea nitrogen, or urea, or BUN, blood urea nitrogen. In fact, let's look at the lab form below. Here's another lab form I stole. I go into hospitals in the middle of the night, sneak in and stealthily steal their lab forms. Now, actually, I go to hospitals all the time and say I'm a physiology teacher and I'd like to see some of their forms because I'm getting used to my class. Which hospital did I steal this form from? It's listed over here. It says Southern California Permanente Medical Group. Who's Southern California Permanente? That's Kaiser. That's Kaiser. Now, there's a Kaiser on Cadillac in West LA. I actually got this one from the one in North Hollywood. All right? So this is listed as blood chemistry. And you'll notice that the very first thing listed when they fill out this lab report is the urea nitrogen level. And in fact, urea, creatinine, and uric acid are the three major organic waste products that are found in our blood and in our urine. All that happens is our kidneys filter it out of our bloodstream and excrete it in our urine. Those are the big three. So in case you're thinking, wow, you know, you're covering all this chemistry, I don't know what this has to do with me, I'm going to be a nurse. Do you think you need to know what these, first of all, these lab reports are, and you think you should know what the 3 major waste products are in our bloodstream and in our urine, that are excreted in our urine. Okay, so now that we've demonstrated what urea is, so coming back here, so we saw on page B10 that urea is a waste product formed from ammonia in our liver, and it's formed from breaking apart amino acids and turning them into sugars. So we see that these ketoacids, that's the name for an amino acid after the amino group has been removed, are formed. Now, back on page, on our big diagram on page B12, so on B12, so the proteins are first split apart into amino acids. The amino group is removed from the amino acid. That's called deamination. It forms ammonia, and the ammonia, NH3, is turned into urea, and then that's ultimately excreted in our urine. It is the major organic waste excreted in our urine. What did we call an amino acid after the amino group has been removed? It's called a keto acid. And this keto acid, or ketone bodies, which include acetone, can then be reversibly turned into an acetyl sugar. This process of converting something that wasn't a sugar into a new sugar is known as gluconeogenesis. You'd say, like, do we have to know that? 100% you do. It's a very important term. Let's break it up. Genesis. Genesis means to make, to form. Neo. Anybody know what neo means? New. And gluco means sugar. So literally it means to make new sugar from something that wasn't a sugar. That's called gluconeogenesis. So whenever the body is breaking down fats and proteins at an increased rate, and we're going to give you examples of that in a moment, whether the body is breaking down fats at an increased rate, as the fatty acids are converted into acetyl sugars, it tends to flood the system so that the acetyl sugars are formed at a more rapid rate than can enter the Krebs citric acid cycle. So some of these acetyl sugars are reversibly converted into keto acids or ketone bodies. And if proteins are being broken down at an increased rate, again, obviously that forms keto acids, because as amino acids are deaminated, that forms these keto acids or ketone bodies, which eventually are turned into sugars or used as a source of energy themselves. Now, what we'd like to do is to give you some clinical applications of this. And so, first off, just before we do, on page B11, so in summarizing on page B11, the catabolism of proteins, so we've summarized that proteins are broken apart into amino acids. The amino acids are deaminated, forming a keto acid or ketone bodies that includes acetone as well as beta-hydroxybutyric acid and acetoacetic acid, but we'll just focus on or emphasize acetone. And what becomes of that amino group is it's turned into ammonia, and in the liver that ammonia is converted into urea. That urea is released into the bloodstream where it can be measured as the blood urea nitrogen levels, and then the kidneys filter that urea out and excrete it in the urine. And in fact, it is the major organic waste product found in both our bloodstream and our urine. The term that we use for when keto acids are converted into acetyl sugars is gluconeogenesis, a very good word. Literally, it means the production, genesis, of new sugars, glucose. So the formation of new sugars from something that wasn't a sugar is known as gluconeogenesis, and that's a very important term to know. In looking at some clinical examples of this, I want to first talk about a situation where people form increased amounts of keto acids, including acetone, and that's when fasting. I didn't write fasting down here, Some individuals do fast during the course of a year, and let's imagine somebody starts to fast. Now, in fact, we have stored in our liver approximately 24-hour reserve supply of sugars in the form of glycogen. So that if you stop eating in the process of fasting, literally, nevertheless, your cells will still be using sugars for most of that 24-hour period. The glycogen would be broken apart into glucose, released into the bloodstream, and made available to the cells of your body. However, as one approaches towards the end of that 24-hour period, as the sugars are being depleted, the body starts to break down increased amounts of fats and, to a lesser extent, proteins as a source of energy as the sugars become depleted. And we've already said that whenever there's an increased rate of breakdown of fats, that forms keto acids. And in fact, these keto acid levels start to rise in the bloodstream, one of them being acetone. And acetone is a very volatile chemical as it is carried in the bloodstream through the lungs. Some of it volatilizes out and into the lungs, and one exhales some of that acetone out one's breath. And acetone, as some of you may know from either a chemistry class or nail polish remover, which at least old versions of nail polish remover used to contain acetone, has somewhat of a fruity smell. So one can actually detect or smell this somewhat fruity smell on one's breath. If one is fasting, one might notice a change in their breath. This idea, incidentally, of acetone volatilizing and exhaling it out, some of it, is very similar to how somebody who is imbibed in alcohol, the alcohol in their blood, as it flows through the lungs, similarly like acetone, volatilizes, comes out in one's breath, and that's the entire basis of a breath analyzer test for determining the alcohol level in one's blood, because the amount of alcohol that one's exhaling out in their breath is directly proportional to the amount of alcohol that's in one's bloodstream. All right, so we've indicated that when one is fasting, as the carbohydrates are depleted or used up, there is a progressive speeding up of the breakdown of fats and, to a lesser extent, proteins as a source of energy. So with this in mind, I want to address the subject of the theory behind either no-carb or low-carb diets. Now, the most severe or stringent of these diets is the Atkins Diet, which is literally calls for eating no carbs. The diets that are not quite as severe would include the Zone Diet, which is a low-carb diet, and there are many, many others. Now, of course, the main reason why dieting doesn't work for most people is they do not adhere to the diet. They don't stick to it. But assuming people do stick to the diet, any diet would probably work, but there is some evidence that these low-carb, no-carb diets may actually provide an advantage. When people want to diet, what they really want to get rid of is fat on their body. They want to reduce the amount of triglycerides in their fat cells and shrink those fat cells, make them smaller to reduce the amount of fat. That's really the main goal for most people who want to diet. Now, here's basically the theory behind it. Let's imagine that you stop eating any carbs. We'll take the Atkins approach. So if you stop consuming any carbs, which is very difficult because carbohydrates include not only sugars, which we use as a sweetener, and it would also include not to eat fruits, which contain sugar in them, or honey. It would also include avoiding all pasta or any other source of starch. Because after all, what is starch? It's just a bunch of glucose molecules joined together. And in fact, when you ingest them, they're just digested into individual sugars that are absorbed into your bloodstream. So that means not eating bread, not eating pasta, not eating potatoes, not eating rice, not eating corn, not eating cake, not eating cookies. That's very difficult to give up those kinds of things. But let's just imagine, in our example, that somebody was going to begin this Atkins diet like right now. Now, as we've already pointed out, you've got about a 24-hour reserve supply of carbohydrates in the form of glycogen in your liver. So for the first 24 hours approximately, the person will still be using sugars as a source of energy. However, after 24 hours, what is the body going to start breaking down at a faster rate? That's right. What the body starts to break down at a faster rate are fats. So as we indicated here, if there's no carbohydrates in the body, what will the cells in the body have to start breaking down at a faster rate for energy? And the answer is fats. By reducing your intake of carbs, you force the body to start breaking down fats at a faster rate. Incidentally, I should have mentioned that in these no-carb, low-carb diets, while they basically call for you to reduce your carbohydrate intake, many of them say you can eat all the proteins you want. You can have all the fish you want, all the eggs you want, you can eat vegetables, you can have all the chicken, all the lean meat, you can eat all the proteins you want, you just have to cut out carbs. And while most of these diets also recommend you don't eat a lot of fats, some even say you can eat the fats, but you've got to cut out the carbs. Again, the reason for this is as long as you continue to ingest carbs in your diet, your body will preferentially use carbs as the source of energy rather than tapping into and breaking down those fats, which is really what most people want to get rid of. Now, we've said that whenever you do break down fats and proteins in a faster rate, it forms keto acids. So in fact, in these no-carb, low-carb diets, there is a rise in the keto acid levels. This is called ketogenesis, or these are known as ketogenic diets. Nevertheless, there's nothing to worry about. There is no record of documentation of anybody ever becoming ill or dying from elevated keto acid levels by being on a no-carb, low-carb diet. After all, most people are not that strict on these diets, and the purpose of a diet is not to be on this very strict diet for your whole life, but to be on it for a few weeks, a few months, simply to reduce the amount of fat that one has on their body. Let's take a look at the second clinical example, and that's where we wrote the development of ketoacidosis in unmanaged diabetes. Let me remind you that diabetes is a very complex metabolic, meaning biochemical disorder. And in fact, there are different types of diabetes. There's one type of diabetes that's really an autoimmune disease that tends to develop in children. And there's another type of diabetes that tends to develop in people as they grow older. And both of them have genetic factors, genetic bases to both types. But they are also affected by, at least the second type that develops later in life, can be exacerbated or made worse by what one eats, and one's weight and so on. But let's keep it real simple. The fundamental problem in diabetes is a deficiency of insulin. Either the child can't produce the insulin at all because their immune system has destroyed their beta cells of the pancreatic islets that normally produce insulin, or in the type where people develop later in life, their beta cells in their pancreatic islets are secreting less and less insulin, or the insulin simply is not affecting the target cells of the body like it used to. So there's a reduction in the responsiveness of the cells of the body to the insulin. In either case, there's not enough insulin. Now, what does insulin fundamentally do? Fundamentally, insulin causes sugars in the bloodstream to be transported into the cells of our body. And as the sugars are transported into the cells of our body, in most cells, the cells will then be able to use that sugar, oxidize it as a source of energy in order to make ATP. In the case of liver cells especially, the insulin makes the sugars go into the cells where inside the liver cells, these sugars can be snapped together into the polysaccharide glycogen to be temporarily stored. So, if there's not enough insulin, what's going to happen to the amount of sugar in one's bloodstream? Well, since if somebody is eating foods containing carbs, containing sugars, those sugars are absorbed into their bloodstream, and these levels of sugars continue to get higher and higher and higher as the sugar accumulates in the bloodstream, because without insulin, it cannot be transported into the cells of the body. This increase in the amount of sugar in the bloodstream is called hyperglycemia. It's called hyperglycemia. Hyper means high, glyce means sugar, emia means in the blood. And the sugar level in the blood becomes higher and higher and higher. And in fact, as the sugar level becomes higher and higher, the sugar starts to spill over into the urine. In other words, normally, as the kidneys filter the blood, all of the sugar that goes through the kidneys is reabsorbed back into the bloodstream. But as the levels of sugar become higher and higher and higher, it starts to be filtered out of the kidneys and appear in the urine. Normally, there should never be sugar in the urine. So in fact, if there's sugar in the urine, which is known as glycosuria, that's an indication that one's sugar level in the blood is very high. The higher the sugar level in the urine, the higher the sugar level must be in the bloodstream to cause that. But in fact, the bigger problem is not the increasing level of sugar in the bloodstream. The bigger problem is the fact that if the cells are not able to use sugars as a source of energy, then what have we learned the body will start to do at a faster and faster rate? And the answer, of course, is the body will start to break down fats and even proteins at a faster and faster rate. As the body breaks down these fats, especially in proteins to a lesser extent at a faster rate, that causes a flood of keto acids, or I should say acetyl sugars that lead to keto acids. As the keto acid levels become higher and higher in the bloodstream, this is known as ketonemia. Again, emia means in the bloodstream. This building up of keto acids in the bloodstream is called ketonemia or ketosis. Now, as the keto acid levels become higher and higher and higher, the keto acids also start to spill over into the urine. In other words, the kidneys start to filter the excessively high levels of keto acids into the urine, and that's called ketonuria. This buildup of keto acids, the real problem that this causes is exactly this. They are acids. And this buildup of keto acids accumulating in the bloodstream leads to a disorder known as ketoacidosis. This is a type of what's known as metabolic or biochemical acidosis. Acidosis due to a buildup of a metabolic chemical due to this metabolism. And of course, we have learned that anytime the body starts to develop acidosis for any reason, the proteins start to unravel or denature. The individual may even lose consciousness as the proteins in the brain cells start to unravel, go into a coma, and even die. So literally, in people who have diabetes to the point where they develop ketoacidosis, it becomes life-threatening. Now, this raises a number of questions that you may be thinking about. One question you might have is this. You might say, well, wait a second. Let me see if I got this right, Professor Fink. You're saying that if somebody has diabetes, they're breaking down fats at a faster rate in their body. Yes, that's what I said. So you'd say, well, if they're breaking down fats at a faster rate in their body, then how come everybody with diabetes is fat? Well, the answer to that is, in fact, not all people with diabetes are fat. Certainly, the type of diabetes that develops in childhood, usually those children are not fat. But even people who develop diabetes later in life may actually, in fact, be thin. But you might say, well, but then, all right, but how could somebody be fat, overweight, if they've got diabetes, which involves breaking down fats at a faster rate as a source of energy? And the simple answer to that is that somebody can be breaking down fats for energy, but if one eats more food consumes more calories of food than one breaks down, they will still gain weight. It's simply a matter of the amount of food or calories that one takes in versus the amount of food or calories one burns or breaks down using it for energy. If somebody eats for lunch a dozen donuts and a half a chicken and French fries and a chocolate malt, sitting all day and not really breaking down much food for energy, even if they're breaking down fats to supply the need for energy, they're still going to gain weight because they're simply taking in more calories than they're breaking down. So that addresses the question that somebody can still be overweight or fat even if they're using fat as the source of energy. Now another facet to this, because of whenever there is increased levels of keto acids in the bloodstream, so in fact one of the characteristics of diabetics, as well as people who are fasting, as well as people who are on a no-carb, low-carb diet, is you can detect or smell the odor of acetone on their breath. Also, I just might mention that it is very common for people who have diabetes to purchase in the drugstore these kind of dipsticks, and what they do is they take these little strips and they dip them in a sample of their urine to determine whether there is sugar or ketone bodies, keto acids in their urine. And they use this color code to determine just how much sugar, if any, and how much keto acids, if any, are in the urine. Again, the higher the levels, the more serious or severe their diabetes is. Now, of course, the way that diabetics try to control and manage their condition is, if possible, they will try to control it by watching their diet and what they eat, watching their weight, and exercising. This is mostly effective for the type of diabetes that develops later in life, known as type 2 maturity onset diabetes or insulin independent diabetes mellitus. But in many ways, the more severe form that develops during childhood, known as type 1 juvenile onset diabetes or insulin dependent diabetes mellitus, in that case, they have to inject insulin. Or an alternative nowadays, rather than injecting insulin, is to wear an insulin pump. Sometimes, especially in people with the maturity onset, they may be able to, if not control it with their diet, may be able to control it by taking an oral hypoglycemic drug such as glucophage or metformin, which increases the effectiveness of the insulin. And the person is still able to produce insulin, so it just increases the release or effectiveness of the insulin in promoting the transport of sugars into the cells.